Right. Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Susie Zhang, Vice President of Regulatory Strategies, will draw upon her 12 years of experience at the FDA focused on physiologically based pharmacokinetic modeling, DDI risk assessment, and model-informed drug development to explain how in silico methods can streamline DDI research and significantly reduce the time and need for expensive study-based clinical trials. Our Business Development Director, Andy Mueller, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. And now I'll hand it over to Andy to introduce our speaker.
Thank you very much, Jasmine. Hello, and welcome to today's webinar, DDI Risk Assessment to Inform Your Drug Label. This will be the first in a series of DDI-focused PBPK webinars that will be offered by Simulations Plus over the coming few months. Thank you very much for taking some time out of your busy schedules and joining us today. As Jasmine mentioned, my name is Andy Mueller, and I will serve as your moderator. I've spent essentially my entire career in pharmaceutical development, and my personal scientific background is in toxicology, bioanalytical chemistry, pharmacokinetics, and PBPK modeling and simulation. My current role is Director of Business Development for PBPK Services, QST Services, and Regulatory Strategies at Simulations Plus. I am pleased to be joined today by my colleague, Susie Zhang. We were thrilled when Susie joined the Simulations Plus family this past September. Susie brings to us nearly 13 years of experience from the FDA, in which she focused primarily on PBPK modeling and clinical pharmacology. During her time at the FDA, Susie oversaw and reviewed many DDI-focused submissions, which truly makes her an expert in what the agency expects of in terms of DDI investigation within the development pipeline. I've prepared a series of questions for Susie, which I hope will reflect what your interests might be. While she takes the time to respond to my inquiries on behalf of you, I welcome you to post your own questions in the chat. We'll do our best to reserve some time at the end of our Q&A to allow questions from you, the audience. In addition, at the end of the discussion today, you will have the opportunity to request a one-on-one conversation with Susie as well. With that, welcome, Susie. Would you like to say a few words about yourself before we get going?
Sure. Thank you, Andy, for the introduction. Hello, everyone. My name is Susie Zhang. I'm a scientist by training, and I've been lucky to work in both regulatory and industry settings, always with a focus on being innovative. I started my career at the FDA's Office of Generic Drugs, which often gets a rap as being about copying or following. I'm really proud of the work my colleagues and I did about 15 years ago to change that perception. We introduced groundbreaking modeling simulation tools that helped shape how generic drugs are developed and regulated. Some of the initiatives we kicked off included using modeling simulation for quality by design, creating virtual bioequivalence concepts, and improving bioequivalence assessment for narrow therapeutic index drugs. Fast forward to today, modeling simulation plays an important role in developing complex generic drugs. After that, I shifted my focus to new drug development, where I had a chance to lead the PBPK program in the Office of Clinical Pharmacology, reviewed numerous submissions at various development stages, and participated in multiple guidance development. Now, as a Vice President of Regulatory Strategies at Simulations Plus, I'm committed to working with teams to solve problems and help drug developers bring their products to market faster and more affordable. I'm also passionate about pushing the boundaries of modeling simulation to continue improving drug development. For today's conversation or webinar, we will be talking about a specific area of modeling simulation, which is physiologically based pharmacokinetic, or PBPK modeling simulation, with a focus on DDI risk assessment from a regulatory perspective. Now I'll hand it over to Andy to start off the dialogue.
Thank you, Susie. Really appreciate you providing us with an overview of your very impressive background and credentials. To get things started, perhaps you could provide us with a history, if you will, as to how the FDA has viewed and interacted with PBPK modeling at a high level over the years.
Yeah, thanks for that question. I love talking about history, especially those I personally involved, because that makes me look experienced. I'll talk a little bit about the journey of PBPK modeling simulation applications in regulatory submission. As you can see, okay, let me do this. I started from like 2008-ish, where FDA started recruiting dedicated staff working on modeling simulation, PBPK modeling simulation assessment. So I would consider those as early PBPK submissions, where the submission numbers were low. During that period of time, FDA staff even did their own modeling simulation. In 2012, okay, let me see. In 2012, there was a pharmaceutical science and clinical pharmacology advisory committee meeting was held to discuss the utility of PBPK modeling simulation in pediatric development. In the same year, FDA published the DDI guidance, where it described when, where, and how PBPK models could be used for DDI evaluation. In 2014, FDA held a public workshop on application of PBPK modeling to support dose selection. In the same year, EMA published a concept paper on qualification and reporting of PBPK modeling and analysis. And after that, I think agencies start seeing a lot of more submissions coming in. In 2016, FDA held a workshop on mechanistic oral absorption modeling simulation for formulation development and bioequivalence evaluation. EMA published a draft guideline on the qualification and reporting of PBPK modeling simulation. FDA began to include PBPK in various several guidance concerning specific clinical pharmacology topics. In 2017, FDA published updated DDI guidance, where they split the guidance into in vitro and in vivo, where in the in vitro guidance included specific guidances or topics on PBPK modeling for DDI assessment. In 2018, FDA published PBPK analysis format and content guidance. And in 2019, FDA held another MIDD workshop focusing on development of best practice in PBPK modeling simulation to support clinical pharmacology regulatory decision-making. In that workshop, FDA presented more case examples where they were able to group a lot of examples into different categories compared to the 2014, where individual cases were presented. So we see more examples in the 2019 workshop compared to the 2014 workshop. In 2020, FDA published draft guidance, the use of PBPK analysis for biopharmaceuticals. FDA also published draft guidance on the gastric pH mediated drug-drug interactions. 2012, ICH published the draft M12 guidance on drug interaction. And 2014, which is this year, the M12 guidance, well, which was last year, we're in 2025, I'm sorry, we're in 2025. 2024, FDA, not FDA, ICH finalized the drug interaction M12 guidance. Meanwhile, they published F15 draft guidance on model-informed drug development. So the most two recent guidances, I would say, are the ICH M12 guidance and ICH M15 guidance, which are relevant to our topic today. I also wanted to talk a little bit about the recent trend in regulatory submissions, PBPK analysis. In the past, FDA has been publishing the submissions to the Office of Clinical Pharmacology. One publication where they summarized the submissions from 2008 to 2014. Another publication where they summarized submissions from 2018 to 2021. So as you can see that in terms of the utility of PBPK in each area, I would say the distributions are similar compared to the two time frame. But if you look at number, the first 10 years versus the next 5 years, the numbers, I would say, almost doubled from 2018 to 2021. If you look at the percentage of PPK submissions in the novel drug approvals, which is the figure on the bottom here, you see some fluctuation, but overall the trend is the percentage is improving. And from 2012 to 2020, from 2022 to 2023, among total 92 submissions, you see about 30 to 40% of the submissions included PBPK analysis. We pick up this 2 years so that we can compare with the EMA submission, which was published in this year, 2025. They summarize 2 years of submission where you see 65 areas of applications. In terms of the areas of applications, distribution-wise, it's similar as reported by the FDA. Majority of the submissions are involving DDI evaluations and there are organ impairment. There are other applications, absorption. In terms of percentage of PBPK in marketing authorization applications from 2022 to 2023, this is based on 95 submissions. The percentage is about 26. So I would say these from FDA's publication, from EMA's publication, we see similar trend in terms of PBPK applications as well as PBPK applications for DDI evaluation. So I hope that addressed your question, Andy.
Yeah, thank you, Susie. I appreciate that. Very nice high-level overview of the history of how the FDA has interacted with PBPK over the years. A few slides back, you mentioned a few recent guidances, specifically the M12 drug interaction guidance and the M15 MIDD guidance. Would you mind giving the attendees and myself a high-level overview of both of those recent guidances, please?
Yeah, definitely. So M12 guidance is a dedicated guidance on drug interactions. It was first published in 2012, and 2 years later, it was finalized. It covers a wide range of aspects of DDI assessment. It's more of a technical guidance where it includes in vitro assessment, in vivo assessment, in silico outside of PBPK, as well as some technical recommendations on how the PBPK models should be evaluated, which we'll also be talking about it later on. So this is more of a technical guidance. If you need some technical details, you should go there. And this is the most recent publication. But if interested, you can also go back to look at the 2012 guidance as well as 2017 FDA guidance, where you will see the science is evolving and the cutoff values change over the time. But this is the most recent guidance. While M15 guidance is more of a general guidance, it included all kinds of model-informed drug development tools, including population PK, exposure response analysis, PBPK, more complex models such as QSP. While from modeling's perspective, these tools or these models become more complex, while in this guidance, it focuses on more on a general principle, like how to communicate, how to define, draft a model analysis plan, how to write a report, compared to the M12 guidance, which is where, in my opinion, there are more technical components. So this is a more general principle guidance. Interestingly, in that guidance, it raised a risk-based credibility assessment framework, which we've seen it appears in a few recent guidances. I wanted to talk a little bit about the risk-based credibility assessment. Why it is important, because model performance acceptance criteria might be related to the risk-based credibility assessment. I listed a few publications here. If we go back again to the history, the first time where they talk about this concept was actually in the 2019 public workshop, if you go back to the history slides. And later on, there was a publication where it talks about consideration of a credibility assessment framework in model-informed drug development potential application to PBPK modeling simulation. In that publication, there was an example using PBPK to help you how you should define the concept of context of use, question of interest, how to define the model influence, decision consequence. These are the questions should be asked early in drug development. And I mean, the assessment may change over time with your model or with your program development, where you accumulate more evidence, you have more knowledge about drug development of specifically your compound. The assessment may change over time. So that was an early publication. And later on this year, where FDA published another guidance on artificial intelligence, you'll see a similar figure there, where the guidance gave two hypothetical examples, how you would define your, how you would assess your model risks. And you see similar in the M15 guidance. So really, this is something evolving. While we're talking about PBPK for DDI evaluation, we look at all the guidance, relevant guidances, not only specifically for PBPK, but for modeling simulation in general, especially when those models become more complex, such as QSP models. We look at them, all those relevant components together. So as I mentioned, this might be related to the model performance acceptance criteria during your model development as well as evaluation. So I hope that gives you a general overview of these two guidances. Andy.
Yeah, that's an excellent general overview of those two guidances. Admittedly, potentially for non-regulators or partners in the industry that are new to in silico modeling and simulation principles, what would you say are the key messages for organizations that are developing novel drugs? What should, at a high level, what should they be taking away from these guidances? What's the message?
Yeah, so I think in my opinion, and also you probably will see that in a lot of the guidances, especially when it involves complex models, modeling and simulation. If you think about how long it takes you to develop those models and how long FDA reviewers have those models for their review in terms of time, well, FDA would like you to talk to them as early as possible. It will help you to refine the strategy, not only modeling simulation, but maybe drug development in general, and also help you define the focus of modeling simulation, types of questions, what types of risks you want to assess, what are the low-risk questions that you probably can address early on? What are the high-risk questions probably need to collect additional evidence? So I would say one important takeaway from those guidance is always to talk to the regulatory authorities early, and also potential collaborators early, such if you need help, feel free to talk to us and definitely can help you to map a strategy moving forward. I think that's important. And the other important thing is to do a lot of the assessment, at least in my opinion, low-hanging fruit early. So for example, by time of pre-IND, you I would expect all the relevant in vitro studies are done so that you can at least assess. But I understand it's difficult. Sometimes you don't have the whole picture of what it will look like in the first in human study. What are the relevant clinical exposure for that specific compound? Those type of things are not clear by the time of pre-IND, but always think early. And the other thing that I wanted to mention is we talk about the novel drug development, but DDI assessment should also be considered when you develop a 505(b)(2) product. So for example, if you expect your exposure will be increased, and then the drug as a perpetrator has this and that potential DDI concerns, then you need to reassess it. It may not be you might not need a dedicated DDI study, but the point is those should be reassessed in the program development stage. Any other?
No, that's
thought?
that's excellent. So, key takeaway is perform assessments early and engage early, right?
Yeah.
So as many of us in the industry know, historically, and today, there are a variety of evaluations that can be used for assessments, in vitro, in vivo, in silico. Can you speak to what might be the appropriate order of events, if you will, in terms of what types of assessments to perform and when? So, for example, should in vitro come before in silico? Are there certain in vitro experiments that are needed to inform in silico? Should in silico be leveraged first? That's the question.
Yeah. Yes. Yeah, definitely. So again, in general, but it could be case by case. From a modeling perspective, you need some input parameters, right? I would expect those input parameters should be generated from well-conducted studies. I understand from early stage, maybe your in vitro studies were conducted many years before it goes to first in humans. So at that point, the studies might not even be conducted at relevant conditions. For example, the concentrations may not be close to the clinical exposure level. But in general, those model input are expected to come from well-conducted in vitro studies. And after that, you have an initial model. You can play with the model to tell you what should be done in the next steps. What are the high risk? We definitely need more data to bridge the gaps where the risks are low, where you can probably use modeling simulation to bridge the gaps. I think the other challenge that I've seen in the past is the communication among different groups. So for example, in vitro studies are done in one group, DMPK, while the modeling simulation is done in another group or clean farm group. So I think it's important for all the stakeholders to come together and discuss those challenges, what was found specifically for that drug. Yeah, that is something beyond, I think, modeling simulation. And then when you move on to phase one study, you have more data. Well, even before first in human, I would say most of the companies may do a first in human exposure prediction. At that point, you will have a general idea in terms of what the clinical exposure will look like. And then while the first in human study is ongoing or the phase one study is ongoing, you'll have more data and then come back to validate the model to make the model more robust and for future analysis. And of course, by the time of registration, which we'll be talking about later, I think you have a question on that. The agency is expecting to have a comprehensive package of what data should be looked at.
Excellent. So a strategy is likely to incorporate in vitro, in vivo, and silico models, and they may inter-inform each other, so to speak, and you're able to, on a case-by-case basis, essentially help partners connect those dots, yes?
Yes.
Excellent. Okay, so when at your time at the FDA, when you were reviewing DDI PBPK submissions informed by PBPK models, can you let us know what you looked for, what current officers might be looking for in a submission? How did they scrutinize a submission? What was a good submission? What was an unsuccessful submission, et cetera?
Yeah, I think that's a I think that's a commonly asked question in terms of what do we need for a successful PBK modeling. From the investigational drugs perspective, so we first develop a model for the investigational drug. Of course, when we develop a model, then these are the input parameters. This includes physical chemical properties, that includes solubility, permeability, drug product properties, that includes formulation, in vitro absorption, distribution, metabolism, or excretion, ADME related studies, like in vitro metabolism SIP panel study, human liver microsome study, and all the relevant drug-drug interaction studies for the investigational drug as a substrate, as well as the investigational drug as perpetrator. So those are the in vitro related studies. And then ideally you would have a mass balance study so that we can confirm that what we put in the model is consistent with what is observed in the clinical study. And ideally, you will have dose proportional study that includes single ascending dose study, multiple ascending dose study. Those will confirm some of the assumptions that we will need for the model, for example, whether the absorption is linear, whether the clearance is linear, whether there are other saturation involved in absorption and clearance. Does the model include all those potential mechanisms from a PK or ADME's perspective? And also additional available PK studies will be needed for robust model validation. That includes pharmacogenomics data. For example, if the drug is a substrate of an enzyme that is substrate for as a substrate of an enzyme that may be impacted by the polymorphism of different genotypes or phenotypes. And do we have PK in patients, and whether the PK in patients and the healthy volunteers are similar? And of course, clinical DDI studies. So those are the basic studies that are needed for a model development and validation. And of course, for DDI evaluation, you will also need relevant, validated perpetrator models and substrate models. I'll give you an example where drug-drug interactions are evaluated. So, for example, we have sorry, this is not the right slide. So how is a PBK DDI model evaluated? For example, we have an investigational drug X, and this drug is a substrate of CYP3A and CYP2C9. It's an inhibitor of CYP2D6. As a substrate of CYP2C9, you may potentially need to consider the impact of genotype. And depending on the fraction of the drug being metabolized by CYP2C9, the genotype or the phenotype may or may not have a significant impact on PK. And then to evaluate the impact of to evaluate the drug as a substrate from a DDI's perspective, we will have a range of CYP3A inhibitors and also a range of CYP2C9 inhibitors. We'll also need to have a range of CYP3A inducers and CYP2C9 inducers to evaluate the DDI potential for the investigational drug X as a substrate. However, in order to develop those CYP3A perpetrator models or CYP2C9 perpetrator models, we'll need to evaluate the sensitive probe substrate models specifically for CYP3A and CYP2C9. And to evaluate the drug the drug acts as a CYP2D6 inhibitor, will need a sensitive CYP2D6 substrate. And in order to develop a CYP2D6 substrate model, we need to have CYP2D6 inhibitor models minimally. So as you can see, to evaluate DDI potential for investigational drug acts, we will minimally need 8 PBPK models as on this slide. But in reality, we will need a range of inhibitors and a range of inducers for each pathway. So this leads to this slide where for the models outside of the investigational drug, which means for the perpetrator models, for the inducer models, and for the substrate models, ideally they were developed in this type of network. So they are cross-validated against DDI studies for different perpetrators, different inhibitors, different strength of inhibitors, different strengths of inducers, different substrates, so that we have more confidence for these models. So those how we have confidence in those perpetrator and the substrate models. So late stage submission checklist. This is again summary of what we've talked about. That from modeling's perspective, we will have in vitro relevant studies, ADME studies, PK studies, additional evidence, as well as simulation design. And from findings perspective, you'll have a report, you'll have to submit all the final modeling files, and also corresponding observation data, all the relevant clinical studies. I wanted to talk a little bit about the reasons for inadequate PBPK analysis or rejection. And this is published in the paper, in the 2020 paper that we published a few years ago. To have these reasons in mind, that will help you to avoid unnecessary stress later on. We can take a look at these reasons. This may not be a comprehensive list, but to give you some idea in terms of what the at least FDA was looking at. And in the recent EMA publication, you probably will find similar summaries. May not be exact the same reasons, but similar summaries. These reasons include, for example, inadequate probe substrate on modulator models. As I showed you before, those substrate and probe probe substrate and modulator models, ideally, they should be developed in a network, cross-validated against different DDI studies, of course, as many as possible. But we also understand for some of the pathways, there are just not good substrate models, or not models, but substrate molecules, or perpetrator molecules there. So it's really beyond the model. That's our understanding of the class of molecules. It could also be lack of appropriate data to support the key assumptions. For example, some of the key assumptions are not included in the model. For example, if your drug is a PGP substrate, but you did not include PGP in the model without providing any evidence, so that could be problematic. But if you provide some evidence saying that it's not relevant because my absorption is so high, or because the in vitro study suggests that Km value is so low, things like that, you could justify that. Well, that will be evaluated case by case. It could also be inadequate calibration clearance pathway for the investigational drugs as a substrate of a potential drug-drug interaction. So this is important because if the clearance pathway calibration is inaccurate, that would potentially lead to inaccurate drug-drug interaction predictions. Could also be a reason that active or major metabolite is not included, which may impact the DDI or the safety and efficacy down the road. So again, if not included, justification should be provided. Could also be inadequate model performance compared to the observed data. So that means the model prediction is far off from the observed data. Key mechanism relating to interactions not included in the model. So again, if not included, justification should be provided. So these key parameters assume should be might impact the DDI potential assessment. And it could also be like confounding interaction from multiple pathways that cannot be separated. So those are, I think, some of the reasons for inadequate PBPK analysis. I think those should be kept in mind while developing those models so that we don't have the stress later on when the model is almost or is ready for regulatory review. I hope that answered your question. I wanted so again, for this DDI network, we are not going to talk about how those models were developed, but we'll have dedicated webinars in March and April to have deep dive into how those models were developed, validated, how those model performance look like, and more to come in March and April. I hope that answered your question, Andy.
Yeah, thank you very much, Susie. Appreciate that. So going back to your time at the FDA when you were reviewing DDI and PBPK submissions, there are different technologies out there, right? So there's some open source technologies. There's some technologies out there that you license. Our technology is GastroPlus. Can you speak to how the agency reviews those technologies? Can you speak to how our technology, GastroPlus, might compare to some of the other technologies that you've seen in terms of submission and submission acceptance?
Yeah, sure. That's interesting. I think, again, a commonly asked question. If I put my FDA hat on, well, FDA will say that they're software neutral. But again, as I mentioned, how those models developed, and after I joined Simulations Plus, I've reviewed all the models that we have developed. I think one of the advantage is that all the models developed in Simulations Plus were mechanistic absorption models. So that's important if you are going to model some of the DDIs potentially happening in the gut, for example, gut transporter related PGP mediated drug-drug interactions, or the gut CYP3A involved drug-drug interactions, and potentially other gastric pH mediated drug-drug interactions, which we will be talking about in May, not in March and April, and later on. So I think that was an advantage I observed. And the other one, as I mentioned here, to develop this network, we're really looking at those probe and substrate models in a network. They're not individual models anymore. We cross-validate them using the same set of parameters against as many DDI studies as we can get for relevant probe modulators as well as probe substrate for the relevant pathways. This probe modulators covers a wide range of interaction potencies from weak to strong. So I think that's those are the advantages I would say based on what I've reviewed in the past few months of our models. And again, if you're interested in knowing how those models were developed, how the model structures look like, what are the pathways have been incorporated, how the models were validated or evaluated, more to come, though in those dedicated or more technical-oriented webinars. But today we're more focusing on regulatory perspective or overview.
Yeah, thank you, Susie. And I know you won't say it, so I'll say it for you. And you just sort of briefly touched on it. But another advantage as I see is that you work with us full time, right? And you have the ability to oversee these modeling efforts. You have the regulatory experience, and you're happy to get involved in these projects to ensure a higher level of submission success. So we'll move on from
Sure.
there. So are there any, you talked earlier on about risk categories. So back when you were speaking about recent guidances, there was an assessment of risk categories. I was always under the impression that the FDA from a DDI perspective was somewhat interested in therapeutic areas or classification of compounds. Wondering if you could speak to that, demystify that as well. So are there any particular therapeutic areas, classifications, et cetera, in which the FDA is paying particular attention to DDI evaluations?
Yeah, I would say no. They
Okay,
treat them
fair
similarly
enough.
from a therapeutics perspective, but in both of these publications, if I remember correctly, they probably both broke down for therapeutic areas. I think the impression that more probably coming from oncology drugs, and that one of the reasons I suspect is because there are more drug development activities in those areas, so more submissions
Okay.
to the FDA in those areas. So you see more cases from those therapeutic areas. But historically, antiviral is an area where there are a lot of drug-drug interaction issues, if I remember it correctly. But in general, I don't think there's any like rank of priority in terms of therapeutic areas, I would say, put that way.
Okay. Personal myth busted. Thank you for that. Appreciate
Mhm.
that. I think that's all the questions that I had prepared for you today, Susie. We were going to mention the additional webinars if you wanted to flip back to that previous slide and maybe reiterate that we're going to have some additional webinars in the series that are going to be more technically focused on the actual models. And we're going to get into a deeper dive in terms of model building, validation, the network of models, et cetera, within the different categories. I think at this time, Jasmine may have had a few poll questions that she was interested in launching. And after that, we can check the chat to see if there's any Q&A that's come from the attendees today. Jasmine?
Yeah, thank you, Andy. We have two quick poll questions. One of them is if you would like to set up a consultation with Dr. Susie Zhang about your company's clinical DDI programs and potential PBPK modeling strategies, and that's just a yes or no. And then we have an open-ended question, and we wanted to see what topics you'd like to learn about in 2025. And I'll give everybody a couple seconds here to answer those two polls. I see some answers coming in, so I'll go ahead and give everyone a couple more seconds.
And as we're answering those poll questions, at present, there's only two questions in Q&A that we'll be addressing after the poll questions. So if you have any specific questions for Susie, by all means, use the Q&A feature at the bottom of the screen. We'd be happy to take your questions. I think we'll have enough time. We should have about 10 minutes left, so feel free to do so.
Okay, and I'll go ahead and end the poll there. And we can start the Q&A. Thanks, Andy.
Sure. First one is from Victor. Can you please clarify what you mean by calibration of clearance pathways? When you discussed inadequate PBPK analyses, do you refer to not correcting for fraction unbound in incubation?
Yeah, that's a great question. I think that's part of it, right? So the fraction, the calibration of clearance pathway, what I mean is, okay, let's still use this example, the hypothetic drug X, right, where you have 3A pathway and 2C9. pathway. What is the fraction of 3A? What is the fraction of 2C9? How did you calibrate that? Probably you will do in vitro panel study and then HLM study, use or even some inhibition study, in vitro inhibition DDI study, and then these will potentially give you different, and then you use intra system extrapolation factors. Potentially, this may give you different answers, and then you will look at include additional evidence such as mass balance study, or maybe some, I don't know, other studies that could give you a better idea in terms of, for example, you will say 3A, maybe 30%, 2C9, maybe 30%, and then the other maybe renal or something like that. So that is what I mean by clearance pathway calibration. There are a variety of methods, and there are even publications talking about where you have more confidence in which method. So those will need to be evaluated case by case, or eventually maybe you'll have a more relevant DDI study for maybe one pathway, and then use the other method for the other pathway. So it will be a variety of tools or data that have to think about for a specific compound, where it will be more challenge when multiple pathways, or especially two pathways are involved. If you expect that each pathway is less than or minimum, probably you don't have to worry about that specific pathway from inhibition perspective. However, from an induction perspective, I would say it's also important to evaluate. So I hope that answered your question. Feel free to type in if any other follow-up questions.
Awesome. Thank you, Susie. Thank you, Victor. From Jason Bake, lab-to-lab in vitro studies can generate different Km Vmax values. For example, famously PGP. How does Simulations Plus deal with or calibrate? And is there a guidance whether in vitro study was performed with adequate sensitivity?
Yeah, so this is a great question. It's known that there are large lab-to-lab variability from the in vitro studies. So ideally, those should be done in a same, like in the same study, in the same lab. Unfortunately, I don't think every company does that, if I remember it correctly. From modeling simulations perspective, I would like to see those studies to be conducted in relevant studies to be conducted in one study. If that's not the case, then we'll have to leverage other available data and to consider the potential variability in the modeling simulation. So again, you have more uncertainty in the modeling and simulation. It all depends on the quality of the data that we have to put into the model. So I hope that answered your question.
Yeah, thank you, Susie, and thank you very much, Jason, for the question. One final question in the Q&A. Again, we have time left, so feel free to type your questions. Don't be shy. This one from Lene Hartvig. Peptide drugs in DDI, question mark. What are the requirements? Full DDI in vitro profile, or how?
Is there a so I don't quite remember on top of my head. In terms of if there's a dedicated peptide drug interaction guidance, you probably want to check the biologics drug interaction guidance. So again, drug-drug interactions are related to the mechanisms, potential mechanisms, right? When we talk about biologics, you probably not worry about too much about the enzyme and enzyme and transporter-mediated drug-drug interactions. However, there are other mechanisms you probably need to worry about, such as the top of my head is the FC receptor. Potential interaction with that, and then that will consequently impact the clearance of the biologics. So again, when we talk about drug, so we talk a lot about enzyme transporter because we come from developing small molecules many, many years ago. We have more experience, so we have more guidance, we have more regulations there. For some of the newer modalities, for example, gene therapy, well, ADCs, it's part of large, part of small, and biologics and other new modalities. We have less knowledge there, but it doesn't mean that DDIs are not there. It's just we'll have to spend time to explore and eventually find potential, well, clinically concerning interactions. But I think peptide drug interactions, yeah, I think you probably would want to check the dedicated biologics DDI guidance if it's there. And also check the M12. I don't quite remember on top of my head if there are any specific guidance there. But what I can do is to write down this question and get back to you after I confirm what I was thinking. I hope
Excellent.
that helps.
Thank you, Susie, and thank you, Lene, for the question. We have another question in the chat from Chris Holmes. Can you comment on experience of CYP3A autoinduction for orally administered drugs and prediction performance? For example, prospective predictions versus modeling of multiple ascending dose clinical data. What's your view on how well mixed profile drugs are predicted? For example, in vitro shown to be a 3A substrate, but also have flags for 3A TDI and induction.
Yeah, that's a great question again. So this is so-called complex DDIs, right? So there are a few examples on top of my head. That the investigational drug from in vitro, the investigational drug is a CYP3A substrate, CYP3A inducer, CYP3A TDI inhibitor. So the question specifically is the experience autoinduction for autoinduction. Yeah, so for this type of complex DDIs, I think you probably need more evidence to support or to provide or to give you model input parameters. For example, we know in vitro, from in vitro to in vivo induction, there might be some under prediction there. It also depends on the fraction of CYP3A being metabolized by your compound. It depends on how you collect your data, whether TDI and CYP3 induction usually happens after a few doses, but competitive inhibition, you probably can get some sense from your first dose. So again, I would say those are complex from purely from in vitro to in vivo, there might be challenges. The utility of modeling simulation is to help you first design the study. If you need to do some study to bring the gap, which are the most needed study that should be done. What are the studies or the scenarios or the clinically relevant scenarios that could be bridged by modeling with some studies and with your knowledge or any evidence collected, not necessarily from dedicated DDI studies, but any evidence from the program development studies. So yeah, so this is a typical case that we've seen in the past, and what we can do is also what I can do is also to give you examples if you wanted to look into how those were done in the past.
This might be a great technical question that we can address in further detail in one of our future webinars as well. So thank you very much, Chris. Great question, very technically detailed and something we'd be happy to touch on in the next webinars in the series. So thank you very much for that. Another question in the chat, which is excellent, from TNU. I love this one. Does the FDA accept PBPK modeling to waive a clinical study and label?
Well, yeah, there are many examples, right? So, directly, there are many examples where PPK model predictions are directly used to inform a dose adjustment with, for example, perpetrator, and then they are in the label. If you maybe go to the publications where both publications, you should be able to find great detail of examples there. So I think I listed the references there. So if you wanted to check the examples.
Excellent. But ultimately, yes, we can absolutely use and leverage PBPK modeling and simulation to issue regulatory clinical biowaivers. So that concludes the attendee provided Q&A session from Susie, and I thank you all so very much for attending today. We really appreciate you taking the time in doing so. You certainly have the opportunity to discuss with Susie and I one-on-one if you'd like to reach out and request a conversation. We'd be more than happy to speak to you in further detail. We have more webinars coming up. Please keep your eyes open for the dates and topics of those webinars. And with that, I will hand it back over to Jasmine to conclude our webinar today. Thank you all very much. We appreciate it.
Thank you
everyone
all.
for attending.
To repeat what Andy said, for any questions we were unable to answer today, someone from our BD will reach out to you with answers following the webinar. We invite you to visit our website to learn more about DDI risk assessment. I put a couple links in the chat if you want to follow us on social as well. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you.
Thank you, everyone.